Okay, everyone, welcome. I think we'll go ahead and get started. Thank you for joining us for this webinar about soybean gall midge. My name is Jackie Pohl, and I'm the Communications Specialist for the North Central IPM Center. Our center is one of 4 regional IPM centers supported by USDA NIFA, Crop Protection and Pest Management Regional Coordination Program. And our mission is to coordinate IPM practices in our region by promoting IPM research, extension, and teaching. Before we begin, I just want to point out the Q&A button at the bottom of your Zoom screen. Please use it to submit questions during the presentation, and we'll answer questions at the end. Now it's my pleasure to introduce Dr. Justin McMechan, who is an assistant professor and crop protection and cropping systems specialist from the University of Nebraska-Lincoln. Today, Justin will present Soybean Gall Midge, Spatial and Temporal Dynamics of a New Pest in Soybean. Thank you for being here today, Justin. I'll turn it over to you.
All right. Yeah, thank you. And thanks for the invite. I'll be covering soybean gall midge, a new species. Some of the core kind of baseline information that's very useful towards the development of potential IPM programs. And there's a number of people on this slide, as you can see, to thank. The big one that has done a lot of the work that you're going to see and a lot of the slides that are in this presentation is from Mikaelson de Silva Lima. He did a master's in my lab and is currently located at Western University in London, Ontario, Canada. Vilma Montenegro, some of her work was related to this same USDA NIFA grant. And so you'll see her as well. And Tainara Pasabam started a lot of the work that led to some of the stuff that you'll see from the 2 master students. Then Tom Hunt, Doctors Tom Hunt and Bob Wright were co-authors on a lot of this work with me. And so appreciate their support. There's the listing for the number for the USDA NIFA grant. And of course, the vast majority of the information you're going to see here is related to that project. So to give a bit of background, I'll probably spend the next couple minutes at least kind of giving you a general background and understanding of soybean gall midge, provide some context to why we're looking at these various aspects of spatial and temporal dynamics of this new species. But it's a new species. The first paper came out in 2019 with its formal identification and scientific name with the help of Raymond Gagne. And so Rosiliella maxima, the Rosiliella genus, contains about 56 species worldwide. The name maxima comes from the host soybean that it was found on for glycine max, and then the level of damage that this insect can cause in the field, which you'll see in the next slide. Some important characteristics, if you're unfamiliar with this species, is that it's typically found at the base of the plant, with some exceptions from mechanical damage, from hail, or anything that could damage the upper parts of the stems. We can find them in those spaces with some of those infestations, or injuries. And then they can kill plants under heavy infestation. So not an uncommon thing. We've seen that since 2018-19 when we started studying this new species, and definitely find dead or dying plants in heavily infested fields or areas. And the infestation's heaviest along the field edge. And that's also quite apparent. There are 2 photos on this slide that I want to direct you to. The first one here, and if you can see my cursor, the 2 closest plants to the bottom part of the slide, those stems are a little bit darker in color than this one further up that's kind of a light purple. That's a sign of the infestation of the insect. And if you peel that back, you can find orange or white larvae under those stems. That's an indication that you have had the infestation. The 3 photos you see here are each from different years of dealing with soybean gall midge in the field. This is a 2020 photo that Nick Tinsley took with a UAV just east of Lincoln, Nebraska, in the eastern part of Nebraska. And you can see that edge effect I was mentioning, where there's a lot of dead or dying plants out towards the middle part of the field. And then a close-up of that. You can see some still living plants among that space. But a lot of those plants, die at different points during the season, depending on the severity of infestation that we're seeing in the field. So now that you kind of understand the context of the severity and presence and occurrence of this insect, here's a little bit on the distribution. So we're currently 7 states. We actually, in the last 2 years, have added 1 state each year, Kansas being added last year and North Dakota the year before. That's courtesy of Janet Knodel and her work, and And so you could see here that in Iowa, and then with Brian McCormick's help down in Kansas, we identified two new counties. That's 6 counties on the year and 1 new state for 2023. No new counties identified in Nebraska, and we're kind of running out of spaces where we have those corn soybean rotations, where it's easier to find this insect in Nebraska. So with a bit of knowledge on where it is, and I guess before I jump ahead, I want to put context to that severity that you saw in the previous slides. Just because we have all these areas highlighted doesn't mean soybean gall midge is causing issues in all these areas. You could certainly find the insect, and that's a lot of what this is detection. But its severity is different, is a different map. So to move ahead and discuss a little bit about the biology and ecology of this insect, we're going to start with where it comes from after we go through the winter. So we have last year's soybeans or our current year's soybeans that are out in the fields right now. If there are signs of infestation or injury, and you're seeing dead plants along the field borders where gall midge was, it's going to fall right into the soil and overwinter. And that's going to lead us right into the research we're going to talk about with Mika's work. But essentially, those adults are emerging from those last year's soybean fields in the spring. We've got quite a number of years of data now, 5 years of emergence data. And you can see that, initially we started kind of in June, mid-June, and now perhaps with an enhancement of how we're placing cages and a bit of understanding, we're really back in that late May, early June timeframe for when we detect adult emergence in the field. And the duration of that adult emergence is a key factor for management of this insect. And so we won't touch much on management in this slide or in this set at all, but if you've got questions, feel free to ask them. But duration is a key issue for how long they emerge from an overwintering site, and really sets the tone for management for this insect. Here's just 1 year. This just happens to be the 2021 season, which will, the overlaps and all the data sources from various projects really overlap well in 2021. So that'll be majority of the focus will be some of that year's data to show what we found. But you can see that they typically peak in about mid-June and then go into July, even mid-July, with adult emergence from overwintering sites. Once the adults emerge, they're seeking out the closest soybeans in the area to infest, and that's where we see those heavy edge effects. And a lot of our looking for new field detection is based on overlapping corn and soybean rotations between 2 fields that are directly adjacent. So you can see how that's an important factor. Once they get into the plant, they infest that base of the plant, like I mentioned in the first few slides. And there's some key things about soybeans when we start looking at them in terms of the development stage of soybeans and their susceptibility. So a V1 stage plant has no real fissures or cracks. If you look at a close-up here, it's nice smooth stem. And then as soon as we hit that V2 stage with 2 trifoliates present, you can see that fissure or crack show up. That's really critical to the infestation. And then if you peel back those areas, you can find them. It looks like we need those fissures or cracks on soybeans to determine or to have a susceptible plant for infestation. Once they're in soybeans and we have that infestation, it's pretty hard to estimate the number of generations that can turn over on soybeans, but a good indication is that we can have 1 full generation, possibly 2 on the current year's soybeans before we get into the fall and winter. That's pretty apparent in this image here. It almost looks like there's 3. This is the 2021 data. 2022 and 2023 don't look as clean as this, but you can see kind of 3 distinct peaks occurring in what would be early July, late July, and August, but kind of as a wash in some years as far as finding those peaks. Okay, so we're going to start straight into the data. That was as brief of an overview as I could provide to give you an understanding. If you've got questions, please post those in the chat, and I'll do my best to answer those towards the end of the talk here. But Mika's first study, and one of our really big questions, especially related to other midges, was we know this midge overwinters in the soil in last year's soybean fields, but we were really interested in its spatial occurrence from the field edge, and then within a soil profile where we might find it. So this little image here is to remind you we're in last year's soybean field, and this is Mika was really good at generating these images of how his data was collected. So this is last year's soybean field and those samples that were taken at each one of those locations. So we pulled an undisturbed core, about 7.6 centimeters by 12 centimeters, And then we divided that core up into several different sections, as you can see here, that were processed separately to determine if if cocoons or larvae were present in each one of those layers, all the way down to 12 centimeters. This was conducted at a number of different sites, and you can see that the sites varied between years, with some sites having an overlap and others not. But we did this in 2019 all the way through 2022. A key point to note that the 2019 data, Tainara Pasabon that I mentioned in the first slide, as well as Dr. Deborah Montezano, those two did a lot of the collections for that particular year. So we really appreciate them kind of pioneering the process on this. And then Dr. Janet Knodel and Kirk Anderson, they're with NDSU, gave us a lot of great training on the process of brine extraction that I'll talk about briefly here in a minute. So this brine flotation, solution flotation method is not uncommon in entomology, and there are lots of good resources like the Doan et al. 1987 that you see below here. I should note, because it explains the amount of work that was done, MICA over the process of all these projects, processed a good 7,000 or more samples. So lots of soil processing, but essentially we're floating out the cocoons. We're initially kind of screening away the soil that's there, getting down to a smaller amount, things that are, the midge size, and then essentially pushing that through and having the cocoons float to the top and capturing those and then counting those is the A, B, C, D that you see on this particular slide. And then we wanted to note that what you're going to see in a lot of the slides that are coming up is that we had 3 life stages we were really accounting for when we say a cocoon, which obviously is just B in this case, but any third instar larvae found in there, as well as cocoons or pupa that were located, are accounted for as present in different parts of the soil profile. So without further ado, we'll jump through the results of that data. So we'll start with the 2019 data. You can really see the bulk of that population is in the top 4 centimeters of the soil surface, but we do have detections 10 to 12 centimeters down, and I don't think anybody's confident on whether or not those individuals that far down could emerge, but a lot of them are right near that soil surface. First 6 centimeters accounts for a lot of what was present. If a core didn't have any larvae in it, or cocoons or pupa through the entire core, we eliminated those in the analysis. So if you see that denominator degrees of freedom jump around a little bit, that's what that result is. It really apparent in 2021, we had very low detections, but still picked up that similar response. A lot of the abundance of the population was present on that 6 centimeters and above, and we didn't detect any below that 6 to 8 centimeter mark, so nothing in 6 to 8 or below. In 2022, numbers are up a little higher, but you can see in terms of the frequency of samples, with positive results, and again a lot in the upper part, but we lost those low parts of the soil profile having any detection of soybean gall midge, but similar responses across the 3 years. So to summarize this study, it's a quick one, but an impactful one. It's the first study to evaluate the distribution of the overwintering generation of soybean gall midge in the soil profile. 75% of cocoons were found in the top 2 centimeters of the soil surface. 96 were accounted for in the top 6 centimeters. And with the exception of 2019, we really didn't detect any cocoons below 8 centimeters. Soil sampling for overwintering sites really wasn't a good predictor. I didn't cover this in those slides, but we actually detected no cocoons at our 2021 Eagle, Nebraska site in any of the cores that were taken from that site, but we still got adult emergence. And so I'll hit on another study that highlights some of the issues of the spatial distribution across the soil surface and where they're located, and just highlights some of the challenges and some of the needs that we have going forward. So you see a number of references on the bottom, and this is just other species that have had studies done on them and how close soybean gall midge is to those studies. So wheat midge, 93.5% of cocoons formed within the top 6 centimeters of the soil surface, so very, very similar. Switchgrass midge, mostly in the top 2.5 centimeters. Burial depths greater than 5 centimeters, delayed or significantly reduced. Swede midge, adult emergence, and sorghum midge, increased a good threefold when its depth was less than 1 centimeter. So all these other midges of different genera, but within the same family, seem to have similar occurrence in the soil profile, which is useful to know that we're finding similar things. I want to highlight that zero data that we got both in some of those years and then within a year and some of those samples that we saw. So we did this high spatial resolution sampling where we pulled 72 cores from roughly a 10 by 10 foot area, a little less than that. But we took cores every 25 centimeters in each direction. You can see the grid there with the C's and L's on it. We did 4 of these grids in the field. We did this in all 3 years of the study, 72 cores per replication. So this was quite a bit of work. We're still sorting out the analysis on this, so there's more to come. But I think even just the graphs are useful for us to understand how complicated this insect is in terms of its distribution. So I pulled out 2 replications, Rep 2 and Rep 3 here, and you can just see how they vary across that grid in terms of where their abundance is. Some grids, very high numbers, or more frequency across the space, and others have very low occurrence and often several zeros amongst that space. So this has been insightful. This was done on the overwintering location, so we haven't done this on the current year's soybeans yet, which we do have a bit of data on that, but in a different format that you'll see a little later. We're going to kind of move past this overwintering discussion. We've talked a lot about the soil depth and profile where they're at. In the process, I should note of that distance from the field edge where we pulled cores at different distances. We didn't see any differences in the data set. That's why it all got collapsed to 1 core for the results, which is interesting considering it's an edge infesting pest, but perhaps that data wasn't pushed far enough in the field to produce a difference. But I think the relevant information really came out of that data set. So we're gonna move over to the current year's soybeans, where they've already moved in and infested those soybeans, and talk about a couple different temporal aspects of soybean gall midge and its importance both on the plant and in the soil. And I'm going to highlight that by the life cycle of soybean gall midge, which has 3 larval instars. 2 of them are white. The third is orange. There's a bit of a gray area as they transition between that second and third instar. But you could see here, Vilma Montenegro's work is showing up in terms of the seasonal larval abundance on plants. So her and Mika overlapped in 2021. And then obviously Mika's work, which focuses on that third instar in the soil profile, as well as silken cocoons or pupa that might occur. That's that 2021 design, a completely randomized design with 3 replications right along the field border. Mika did a more complex design in 2022. But you'll see the amount of data collected would have made it nearly impossible for Vilma to process all those samples that quickly. We'll cover Vilma's data first on the presence and abundance on soybean plants, and then we'll move to Mika's soil data, which we have an overlap in the sites, and I'll point that out for you. So there's that 10-foot or roughly 3 meter by 9 meter or 30-foot long plots arranged along the field edge. What we were doing was going along sampling 9 randomly selected plants for the occurrence of them on the plants, and then taking 3 stems from each plot for Vilma's work every 3 days during the season. Each one went into a separate conical tube, and then we counted all orange and white larvae under a microscope or stereo microscope. This is the frequency of infested plants. On the x-axis is the time, on the y-axis is the number of plants. And you could see that once we really get gall midge in the system, we float anywhere between 60 or 70 so odd percent and 100% of plants infested through the majority of the season along those field borders. This is a heavy to moderate infested field, so it's apparent it's infested from the field edge. If you're wondering what lower infestations or presence might mean, we probably wouldn't see that high of a frequency in those types of fields. The next one here is a little bit more dense, and we've kind of broken that up with some black bars that indicate the averages over those periods. On the x-axis is that same time frame. Y-axis is the number of larvae per 3 plants from each plot. So that's the sum of those 3 plants, sum of larvae on those 3 plants. And you can see first adult emergence of this site was June 8th. By the 14th, we had detection of larvae on those plants. and they remained relatively low through the June period and into the first part of July, mid-July. And then they peaked in late July, early August. And surprisingly, this was similar across all the sites. The 4 site years that Vilma had was that peak population mid-July or late July and early August. And then it's a steady population decline on plants, but they'll stay on the plants all the way through until almost harvest. This was a 2.8 maturity group that this was conducted on. So pretty much the day we harvested, she took her last samples and managed to pick up a 0. But those individuals that are on there are pretty dried up and almost look dead, although we didn't check their viability. Very few present anyway at that point in the season. We'll move to Micah's data. He has 2 years of studies. He has one similar to the design we just saw, which is right along the field border. And then given the potential differences with distance into the field, he added in additional distances in the 2022 field season and started sampling around June 23rd and went till October 12th with weekly samples. So another pretty dense data set. Took 7 random cores from each one of his plots and made his plots a little larger, and then composited those for a response. You could see obviously the reason to do that given the issues of the spatial distribution of this insect in the field. And we went out to about 18 meters or around 60 feet with this sample set. We just used a simple core sampler that stacks these little cores pretty easily and makes for quick sampling. A shout out, I guess, to Bruce Potter on that because he was using this up in Minnesota. So we quickly adopted a much easier system for sampling than our large core sampler. All the graphs are going to look very similar for what you're going to see for the next several slides, which is the abundance of cocoons being those larvae, cocoons or pupa that would be present in the soil, those weeks across the x-axis and abundance of those cocoons on the y-axis. We're going to hit 2021 and 2022 data back to back here and just watch for the differences between locations within a year and then between years on what we saw. So we've got 2 sites here. We named Memphis and Davie. The Memphis site would be the overlap with Vilma's data. So you can see that 2 weeks following where Vilma's larval populations peaked really matched the cocoon populations that were present, at least for a brief period of time there about 2 weeks after that peak population occurred. And then you can also see kind of 3 more distinct peaks in her data set, or in Micah's data set that follows that. The contrast is to the Davie site, which had really 1 peak, pretty low populations through the season, and then 2 kind of smaller peaks towards the tail end of the season. What is the real difference between these 2 sites? Observationally, the Davie, Nebraska site had a far greater amount of pressure earlier in the season, and we nearly lost all the plants from the edge of the field where the plots were in earlier in the season. So there's very few plants to pick from. Vilma was able to retain her data because she sourced only live plants and not dead or dying plants, which is in her thesis if you're interested in the details. But Micah was sampling the soil around these spaces, and so not uncommon or not an unusual expectation that populations would decline if we lost a lot of plants along the edge of the field. Memphis had heavy infestations, but plants in general, there was more live plants that were maintained in that area. And they have some injury scores that Mitchell Helton developed, and I was part of that publication. So they use that. So that's a good precursor for the number of dead and dying plants that were out in these spaces. As we move to 2022, we have the same Memphis site. We're just across the road. We have a new site, Walton, but you could see how different the 2 years are immediately. If I just switch back and we quickly look at 2021 to remind ourselves what it looked like relative to what that next year looked like, which is we have very low populations early season. That peak on mid-July that we saw in previous years, we saw it in late July or early August where we started to see a real population show up, and then they just kind of built through the remainder of the season. What's interesting is both years and all sites that we conducted this on, we saw a decline in the populations as we moved into late September in the soil. But obviously larval populations are on a decline But it was interesting to see that decline also occur in the soil where we were sampling for them. So a bit of a summary on this. Larvae were detected on plants within 6 to 9 days for Vilma's study. So you saw that adult detection in that graph. And then, on the 8th and by the 14th, we had detected larvae present on plants. There's a lot more detail in those studies in terms of the occurrence and abundance of white larvae versus, pigmented versus non-pigmented. That's how we kind of divided those categories up. Wasn't always accurate between all the sites. It sounds pretty, sure based on the number of days, but the cages that we're using are only as good as where we place them, and there's certainly some work to be done. And the development of a pheromone is underway through work with Dr. Ian Kesey here at the University of Nebraska. He's in the School of Biological Sciences. So he's looking for a pheromone, and that would have been very useful for a project like this. Peak larval abundance, as I mentioned earlier, in late July for 4 of the site years of the study. So that peak varied on how intense it was, but it typically occurred in late July, early August. And then the peak abundance for presence of soybean gall midge in soils was quite a bit more variable between sites and during the season. So we had multiple peaks in 2021 and this gradual increase that we saw in 2022. But in both years, we saw that mid to late September decline, which was pretty interesting. Okay, I'm going to move to a pretty different look and view of soybean gall midge in terms of its ability to move from an infested plant and away from that plant out into an adjacent space around it. And we took a couple different approaches to this study. I'm just going to talk about the 2022 data that we have. And there's lots to sort out on this graph. So you're going to see this for a minute or two while I kind of highlight some of the details on it. But we have these 2 round areas of soybeans that were planted in the center of what would be a 15.25 by 15.25 meter area along the edge of a field with no soybeans adjacent. There's just a road here. There's soybeans across the road that were the source for infesting these plots, or these little round soybean patches. And then what we did around each patch is we took 8 different directions from the plot. At different distances, we pulled 2 adjacent soil cores, trying to determine what the spread was around those soybeans for how far the larvae could move from a host before going into the soil. And so you can see that. And then we have this simulated canopy of a non-host. This was buckwheat. So buckwheat's not in the same family. Fabaceae, Fabaceae is soybean. It's a Polygoniaceae, so that we didn't expect any hosts. This host would carry soybean gall midge, and it appears from that work it doesn't. It would break a lot of rules and understandings that we have about the genus Rosiliella if we were to find it on another family of plants. We planted this non-host to hopefully replicate the canopy of what soybeans would be. So if we see differences in movement between these 2 different patches, that would be an indication that canopy might be a critical factor. Mostly we were worried about their survival moving on the soil surface with high temperatures potentially. So McMechan monitored the symptomatic plants from these round plantings of soybeans on that bare ground, as well as buckwheat that was around the plots. And you could see that we obviously got a pretty heavy infestation. And interestingly enough, the observation that we noticed partway through the season was how much harder hit soybeans were when they were in buckwheat. So there could be quite a few reasons for an interaction like that. But the buckwheat patches definitely where soybeans were present took a far larger hit. We took one composite soil sample containing 2 cores per distance, as I mentioned earlier. And then those cores were 6 centimeters by 5 centimeters. There was no need to go all the way down in depth when we knew the majority of the cocoons were retained within that top, 4 centimeters or so. And so you can see that overhead view of how the samples were taken around each one of those patches, 3 reps total. We took 2 different sets here. There's no way to pull these cores, run them into the lab, process all of them, and know you have, a lot of larvae present or cocoons present. And so we took 2 back to back, roughly 11 days apart, to hopefully capture what would be a peak for occurrence. McMechan was monitoring the populations on the soybeans and definitely noticed some declines that kind of triggered that process. We were hoping to pick them up with that. We would have went later in the season, but the soybeans really had mostly died earlier than that peak larval abundance and expected peak cocoon abundance that we saw in both Vilma and McMechan's work. So this is the graph you're going to see. On the x-axis there, we have different brackets of that 6 centimeter diameter. So 0 to 6 right next to the soybean plant. As you can see, it's kind of grayed out in the background. And then 10 to 16 centimeters at intervals that are nearly doubling all the time, with the exception of 300 and 600 centimeters away from the plant to see how far we could detect them away from that host. This is some of the first material you'll see on that. I'll mention the 2019 study, which was the greatest movement we observed. But this initial study shows the right adjacent to those plants, the 0 to 6 centimeters. We could detect them at 10 to 16, and then out to 80 to 86 centimeters was the furthest they were detected in this study. If you kind of, put a box around where we saw activity, that was definitely the greatest level of activity relative to all the other samples that were around there for what was taken. So definitely a significant number. And then I'm just waiting for my slide to advance here. I think these are a little bit heavier of a slide. Yeah, so when we look at the 0 to 6 centimeter comparison, definitely significant relative to all other samples that were taken. So that was the majority of the abundance occurred within that space. If you take those first 2 spaces that were there, that encompasses a good deal of the data. And it's really about 95% of everything that was there was found within the first 16 centimeters of that plant. So pretty interesting study. We were in part pursuing this type of data because we had put cages off the edges of soybean fields, and we were detecting a very low number, but some soybean gall midge in those vegetated borders along the edges, which raised concerns about other hosts. But I think this data highlights the potential they could get into those spaces. And that box is just to indicate the furthest distance that McMechan had observed soybean gall midge from the source. So the distribution pattern of soybean gall midge was not affected by plant canopy, hence the reason you saw everything kind of come together, which was intriguing. We were anticipating seeing some differences. Greatest abundance of larvae was within the first 6 centimeters. 95% were found within the first 16 centimeters from the plant. And his furthest detection was about 86 centimeters away. The 2019 study was just a 2 rows planted and then parallel sampling away. And we detected soybean gall midge at 300 centimeters. So that's a good 10 feet. That's the furthest samples detected. Very low occurrence out at those spaces. And there was some lack of presence in between those, but an indication they could move as far away as 10 feet or 300 centimeters from the source plant. So in terms of the significance of this work, the depth, the majority of larvae and cocoons or pupa were found in the top 6 centimeters of that soil surface. Spatial distribution of larvae across the soil is highly variable. Larvae on the plants were detected about 6 to 9 days after adult emergence. And that difference in larval abundance on plants and soil was observed throughout the season. There are some connections, like you saw in that 2021 Memphis, Saunders County site. But I think it's differences in the number of plants that are alive. And then obviously that decline is very interesting, could be related to the number of surviving plants and especially post-harvest, as everything's kind of cleaned off, maybe a harsher environment for those midges if they're so close to the surface, but really no studies done on that yet. And then the majority of larvae, 16 centimeters, but up to 300 centimeters based on a couple of years of data. So I wanted to leave lots of time for questions. There's plenty of people to thank McMechan and Vilma for their MS thesis work, generate a lot of this data. Tainara Passabaum, who's at North Carolina State University and working on her master's, and Dr. Deborah Montezanos with Corteva Agri-Sciences, but was a postdoc in my lab. Thanks to Dr. Janet Knodel and Kirk Anderson for their help with the soil work, and then Drs. Tom Hunt and Dr. Robert Wright. And then, of course, my lab here at UNL assisted with a lot of this work. Mika would still probably be out sampling had it not been for all those people's help, and I know he greatly appreciated that. Thanks again to the USDA for funding this work. I think it's given us a lot of insight into potential management practices and an understanding of peak populations during the season. And then obviously the University of Nebraska that indirectly and directly supports a lot of this work. So with that, I'll take any questions. I'll put a quick reminder in as the questions are being put together, but there's the website, soybeangallmidge.org. It's getting a little quieter since we're out of the season, but that's where we post a lot of information on first adult emergence and key management considerations. And so you can go to that website. You can also register for alerts that'll provide you with some information when it emerges in the field. And then we'll be doing that again here in 2024. So with that, questions?
All right. That was very interesting. Thank you so much, Justin. Just a reminder to everyone to put your questions in the Q&A at the bottom. And as we're waiting, I have one question. So I'm hearing you say that soybean gall midge appears to be mostly in the top portion of the soil. Have any studies looked at tillage as a possible management practice?
Yeah, a pretty obvious direction with them being so close to the soil surface is what role would tillage play? We've done a number of studies, and Ben Colby with Dr. Aaron Hodgson's in Iowa, he did his master's thesis on that work. You could guess how difficult it is to do that type of work given the spatial distribution of this insect and its low occurrence in overwintering sites where we target a lot of that tillage work. And so we've seen some numerical reductions in the number of adults emerging with tillage practices, those being fall tillage or spring tillage or a combination of both fall and spring tillage. Nothing has wiped them out in terms of seeing a really stark contrast between those treatments. I know the grower at the Davie site that I have, I'm working with, he's frustrated enough with soybean gall midge. He's bought a moldboard plow and is going to moldboard plow the edges of his field. So if that doesn't, indicate the level of concern experienced by some growers, he's willing to work with us to lease spaces so we can understand the impact of moldboard plow relative to other practices. But some growers are certainly contemplating and doing field edge tillage treatments against gall midge.
Thank you. Moving on to other people's questions. Was there anything unusual about the weather in 2021 that led to the short period of emergence?
Yeah, every year is unusual. I don't know what's usual anymore. It seemed like we shifted in 2020, and that shift could be a variety of things. Growers changed up varieties that we were using, and perhaps that had some impact on the population. We've experienced some dry spells in the spring for a couple of years now, and some of our general observations on those drier springs is that they don't help with emergence for soybean gall midge, a good thing for growers. And we've done artificial rainfall studies during the season. And those artificial rainfall studies point to the fact that precipitation events do lead to increased emergence of adults. So a dry spring could limit their ability. For a couple years, although it hasn't been really consistent, is any rainfall event we get during the season usually is followed by a large amount of emergence of adults in the field. So that may soften the soil surface and help with their emergence. But there's a lot of things tied up in that scenario of understanding, including the entire winter that might be compounded on what we see in the spring, how harsh that winter is. I know Minnesota and Phelan Anderson with Dr. Robert Koch, they're doing work on supercooling temperatures of soybean gall midge, and the initial results appear that we won't lose many during the winter. They're pretty hardy in terms of their survival. So great question. I don't know if I gave much good answers, but I gave you some things to think about, Kayla. So.
Yep. We've got great work, Justin. And is injury on a soybean stem required for gall midge infestation? And is it the same across other midges?
Yeah, good question. Okay, is it required? No. We have those natural fissures or cracks at the base of the plant where they tend to get in and start. And so that naturally occurs at stem expansion and that outer epidermis and layer cracking. Injury certainly increases infestation. Natasha Umezu has a lot of really good data we have a simulated hail machine that shoots ice at 170 miles an hour. So we've done some work on that. And certainly that mechanical damage that occurs, they show up in those spaces. Not all, but we've seen larval number increase as much as 5 times on a single plant relative to just fissures and fissure presence. But there's a lot of space for understanding. We did see this past year interactions between 2 species or pests of soybean, Decty stem borer, and some of it's feeding on petioles and getting into stems and then burrowing into the main stem of the plant. Where it initially fed, we saw gall midge follow it in. And that was pretty interesting and widespread because we were hearing about that in Iowa as well from some consultants. That's not new to soybean gall midge. In previous years, we've seen weevil on sweet clover, another host for it. It tends to follow that weevil into the plant. So it uses, I guess, other insects to obtain and get in. For relationship to other midges, their infestation of the points of plants are very different. So some are infesting the heads and other places on the plant. I'm not aware of their need for mechanical damage. So I think that mechanical damage is more of an exception to the rule for soybean gall midge. But I won't speak for all midges. There's too many. So there certainly could be one out there that thrives on that type of system.
Yeah, that's really interesting. Next question. Has there been any fungal pathogen associated with this insect?
Yeah, excellent question. And thanks to the Nebraska Soybean Board, because I'd have no answer for this question without their support. And you probably noticed their logo showed up a couple times during the slides here. They've supported a lot of work on soybean gall midge. We tackled this early on, right after this midge showed up, because we had indications from a related species, Rosiliella theobaldi, the raspberry cane midge in Denmark, Poland, that there could be an interaction with fungal pathogens. And so knowing that we might be dealing with two discipline issue and for growers, possibly 2 directions of management, we thought this was important to tackle right away. We found a lot of fungal pathogens on soybeans that were infested with soybean gall midge, but we don't know the chicken and egg scenario of who showed up first and who's benefiting from who, or if there's any benefit or just co-association, or co-occurrence on those plants. So that there's a lot of work still to be done, and we have an upcoming grant that started here in October to hopefully dice that out a little bit better. But what we did was create a kind of a long catalog of things we found with the 2. We know from that work that we don't need fungal pathogens present for infestation. So that was probably one of the most critical things to come out of there, and that we didn't see like enhanced problems occur with fungal presence. So we did some elimination with fungicide applications, basically doing a wipe out all disease possible with some unconventional practices. Still saw soybean gall midge, still saw very significant populations, and no real yield benefit from that fungicide use. So we got a few things out of the way that were useful to growers, but more room in the upcoming grant to explore that a little bit further.
That sounds great. Well, thank you for all the wonderful questions. Oh, I see another. Do adults cause injury?
Excellent question. The adults are very short-lived. So I think Minnesota has a survival study going on adults. They don't live very long. So that's one part of that whole scenario. They do not cause direct injury or feeding. They don't have the mouth parts to feed. They're more seeking out water. And then laying those eggs, because 5 days is kind of a maximum survival under ideal conditions. Typically probably down around 3 with the harsher conditions that we see. So they're laying eggs. That's the critical part. But no worries on the adults causing direct injury to soybean.
One good thing, I guess.
One good thing.
Okay. We've got another question. Have there been any variety and seed treatment trials?
There have been a lot. So we have done with work with George Graff, our soybean breeder here at Nebraska, 4 years of trials looking at the U.S. germplasm. We've tested 765 accession lines for 3 years. It's a tremendous amount of work to categorize all those. We saw across those years of studies 100% infestation. So all varieties were infested or accession lines. That's the bad part. The good news is we saw varying levels of larval populations and then less injury on some of those varieties. And those have been carried forward, and we're now at 40 accession lines that we're testing. We're still seeing that, good survival. But we're not going to get rid of gall midge, even if we come up with some of the best of that germplasm that we've seen. I see varietal differences with growers. So from our commercial side, just two varieties I have to plant as part of different studies, you can see quite a bit of differences. The trouble with all of that data is gall midge has a choice. And when we take away that choice and a field's planted to entirely one variety, we learn a little bit different of an understanding. So there'll be some caution moving forward. I've encouraged my industry partners to work on submitting a list of varieties they would like to test to provide a screening, much like we do for diseases, so that growers could start off the season on the right foot in terms of some potential tolerance. On the seed treatment side, yes, a lot has been done. We have tested every available commercial seed treatment for soybeans over a number of years and seasons and at different rates. Those results have varied. In some cases, we saw a pretty good response early season, a reduction in larval number. In most, if not all cases, it was hard to carry that to differences in yield at the end of the season. We might see some little numerical changes, but nothing that was statistically significant. I don't think anybody's surprised by that outcome. It would be a lot to expect of a seed treatment to carry on an insect that arrives at V2 and has the capability to infest for the entire season. So there's opportunity space there for an integration of different management practices, cultural control practices. It may have a place, but on its own, it doesn't make much difference. And I guess I'm speaking purely to just seed applied, not the in-furrow, foliar, or granular at plant versions of things with those.
Yeah, that makes sense. And then this will be our last question. Could climate change be to blame here? Why is this pest becoming a problem now?
Yeah, man, that's a big part of what we're trying to figure out. We have, I think we're running a good 36 studies a year. One of those is a survey where we go out and we look, and that's a Nebraska Soybean Board funded project as well, to determine the occurrence and severity of this insect in the landscape. Why am I talking about that? Because part of that data set is linked to weather and some of the differences in weather that occur across the state. And if anybody's been in or around the Midwest this year, we've noticed a lot of variability in the landscape as far as precipitation and temperatures and other things. So that data set probably gets to the role of climate change. I don't know if it's to blame, but the role it might play in mostly weather, I'd say, because that's a shorter kind of temporal way of looking at this. I don't know if it's as much to blame. Certainly something environmental happened. For us to sit on an insect that could have been around 2011, 2012, we saw orange larvae back then. Nobody collected any samples. We don't know if they're soybean gall midge. But if we take the assumption that that was soybean gall midge, it suddenly became a more widespread problem in really 1 year in 65 counties. It suggests some environmental trigger pushed them to be from a late season occurrence on soybean to something occurring in June. And now it's kind of embedded into soybeans as an early season pest with potential. There's probably something like that. The things we don't know are about the host relationship on other alternative hosts like sweet clover, now lead plant we identified as the host. That's the first native host identified for soybean gall midge, which was literally 6 to 8 weeks ago that we learned it was a host. We've got alfalfa and dry beans. So there's a complex system there. Whether abiotic factors are there somewhere, how big they are relative to those other factors is something to be determined.
Thank you. And thank you everyone for your engagement. Our next Pest in Progress webinar speaker will be Lene Just from Michigan State University. She will share highlights and impacts of the 2018 to 2022 USDA NIFA North Central Integrated Pest Management Center. And that will be held on Wednesday, November 15th at 1:30 p.m. Eastern and 12:30 Central. We hope you can all join us again. And you can visit our website, ncipmc.org, to subscribe to our newsletter and keep up with these future webinars. And Justin, thank you again.
Thank you.